11 o'clock, the first Sunday of the month, we are at Prithvi Theatre with Chai and Wai. This is a very special session because we are part of the Prithvi Theatre Festival as well. So the Prithvi Theatre Festival begins today, and this is one of the first sessions that's happening. And it's a pleasure to welcome Dr. Mayuri Rege, who will speak to us today about the little RNA who could. Not the little engine who could, but the little RNA who could. So something about RNA and biology, I guess. Before I introduce Mayuri, let me just, in case somebody's watching for the first time or has come for the first time, Chai and Wai happens on the first Sunday of the month at Prithvi Theatre or Prithvi House here. The third Sunday of the month at Ruvkun College. And if there is month has a fifth Sunday, then we are live from a lab in TIFR. It's an online only session. Okay. And with that, we are actually now into a series of sessions that are going to look at the Nobel Prizes in science for this year. And we're going to start off with the Nobel Prize in Physiology or Medicine, which is being given to these little RNAs that you hear more about. And to tell us about that, it's going to be Mayuri. Mayuri did her bachelor's in Mumbai, Ruya College, I think. After that, did her master's in malaria-based work at TIFR. Went to the US, did a PhD at the University of Massachusetts Amherst, which is going to figure in the story, and I think I'll let her tell you that. And then did a postdoc there, and eventually came back to Ruya College as a assistant professor there. And we were very lucky that we could attract her to TIFR and HBCSE. She joined last year, and she's a great storyteller. So you're going to enjoy this session. Those of you who are watching online, please remember that the only way we can get your questions and comments is by the chat. So please put all your questions and comments in the chat, and we will take a look at them in YouTube. Okay, those of you who are here, feel free to raise your hand and ask questions. Thank you all for coming. You not only get to hear Mayuri, you also get chai. Online guys, you have to make your own. And of course, after the chai break, you will have the opportunity to ask her a lot of questions, and there'll be a lot more interaction. So, thank you again for coming. So with that, I'm going to hand it over to you. Yeah, it's a good idea to keep your cell phones on silent and all that, You're at Prithvi Theatre, you know what to do. Okay. So on that note, let's get started.
Thank you, Arnav. And good morning, all of you. Thank you for making all the trip from wherever you're coming. So I'm really excited to speak to you today about a topic that is current because of the Nobel Prize, but something that has been close to my heart because I've been studying and working in the field of molecular biology for almost 10-15 years now. So
you need to start the section. Now you need to share screen.
Yes, thank you. One second. Is it working?
Yes.
Yeah? Okay, great. Okay, so let's get started. Why we are here, right? So this year in 2024, the Nobel Prize in Physiology and Medicine was awarded to these two gentlemen, Professor Victor Ambros and Professor Gary Ruvkun. They have been credited with this very, very astounding discovery where they have discovered a new type of small RNAs called microRNA, and what its role is in post-transcriptional gene regulation. So I don't know how many of you are from a biology background. Can you maybe give me a sense? Maybe you want to, when was the last time you got into anything related to biology? 10th grade. All right. So this is excellent. Yes. So I think to understand what the significance of this discovery is, we need to understand some of these words, right? So we're going to break this down into a little bit of the introduction, and then we'll get into the discovery. So, right? So when you think of biology, you think of cells, right? Cell is the basic unit of life, so on and so forth. But have you actually ever seen cells under the microscope? I don't know if you recognize this. These are onion cells stained with something blue. Can you tell me what you see? You see a nice packed layer, right? And there's some dots. This is actually the nucleus. And this is what we know as the cytoplasm, the stuff around the nucleus. Let's not go into more detail. This is a plant cell. This is a animal cell. I know there are some teachers in the audience, they're like, ye bahut khali lag raha hai. Never have I ever seen the picture of a cell so empty, right? In fact, it is not. It's just that what we can see with a light microscope, this is what we can see. But actually, if you do electron microscopy and so on and so forth, this is the standard picture of a cell that you will see in a textbook, right? So there are a whole bunch of organelles. I am not going to get into that, right? As few names as possible today. So what I need you to remember is just this blue dot, which is the nucleus. Nucleus में क्या होता है? DNA, right? DNA, the hereditary information that allows, father to pass on his genes to sons and mothers. Well, we'll not get into that today, the inheritance part of it. But what I want you to remember is there is stuff inside the nucleus called the DNA, and there is stuff in the cytoplasm which will interpret that DNA, make it into proteins. Okay, that's it. So these are what we will call as organelles. But now we are going to go deeper. We are slowly going at higher and higher magnification. So if I go further in magnification at the molecular level, what do I see, right? Maybe some of you are familiar with this, but if not, let me remind you. At the molecules, the ones that are very important, we just talked about, which is the DNA. This is the beautiful helix that we know is responsible for passing on DNA, for passing on genetic information from one generation to the next. We know that this is how this molecule has gotten this property because it can replicate itself. It can form more copies, right? Now, this itself is not enough to make life sustain, because if you have DNA, that's kind of a static molecule in that way. So you need to read it and you need to do some more things to it. So these two processes are going to be kind of key to what we talk about today. These are very, very core concepts in what we call as molecular biology. Okay, this happens in every single cell, every single living cell. So let's understand what is happening here. DNA is being made into another molecule called RNA. This one looks like one strand. This is a double helix, right? So right now, that's all we can remember. Let's not go into further detail. And then RNA, this new molecule that we have just made, has somehow gotten translated or has gotten converted into a protein. Protein, everybody understands, I think, right? We eat protein to get strong, so on and so forth. So proteins are what we are very familiar with as things, the machines that do stuff in our cells, right? We have a whole bunch of proteins that act in various different levels in the cell, and we'll briefly touch upon that, right? So any questions, please interrupt me and I'm happy to answer them along the way. The main thing is when we are, I don't know if you've learned about this in your 10th standard class, but this is called the central dogma. And what that means is, because this process, this entire process happens in every cell, it is absolutely critical to understand how it works. But what is often less appreciated in textbooks is the regulation. Matlab, it's not just the fact that I have DNA being made into RNA. How much RNA? When do I make a specific RNA? So on and so forth, right? So there's this level of fine-tuning that is absolutely essential to give us something more. And what is that? So let's take a human being. Let's take any one of us. Every single cell in our entire body has the same identical DNA. The DNA is the same. Barabar? But then how do we get all these different cell types? I have skin, I have hair, eyes, heart. Have you thought about this? Right? So there is something else beyond just the DNA sequence that specifies cell identity, if you want to call it that. Okay? And that is what I was alluding to earlier. This is the first difficult word in that Nobel citation, the gene regulation. मतलब क्या कि even if I have the same DNA अलग अलग सेल में एक ही DNA है, पर फिर भी जो रिजल्टिंग सेल आ रहा है उसमें से वो अगर अलग है, so that DNA is not being read the same way in every different cell, right? And that we know from a tremendous body of work is largely done by proteins, right? So let's go back. This family of molecules, proteins, are what I'm showing you here. Okay, so what am I trying to show you? From the same person where the DNA is identical, if I take a cell from the liver, a liver cell, and if I artificially add these proteins, okay, I can see in a dish that I will make a neuron. इसका इसके साथ कुछ संबंध है क्या? Not really, no. A liver cell should never have to become a neuron in a normal state. But what this shows you is it is sufficient to give it that property if I add specific proteins. Proteins are regulating, they are controlling this particular cell, turning into something it normally would not have. Okay? In the same way, another cell, so DNA same hai, is cell or is cell ka DNA is identical, correct? I put some other proteins. So for example, you might have, people call this a cocktail, there's nothing very fancy about it, right? Protein cocktail to make neurons. Different protein cocktail to make a pluripotent stem cell. Maybe some of you recognize this. You remember Shinya Yamanaka? There was this whole, in 2006, the Nobel Prize was given for this particular process where somebody decided that they can take skin cells from a patient or from a normal human being, and you can actually convert them to pluripotent stem cells. These are cells that can, in principle, be converted to any cell type in the body. ये तो उल्टा ही हो गया, right? A cell that is defined, a skin cell, is now being turned into something of endless potential almost. Okay. How is this process happening? Again, some proteins are acting. So the reason I bring this up is because this process is so well studied and so well known that it will now become clear why this new discovery was so shocking. Okay, so the central dogma, which is true in all cells across life, it says that DNA is made into RNA and RNA is made into protein. And normally, this is the functional entity. मतलब सेल में जो काम है वो प्रोटीन कर रहा है. DNA, hereditary, yes, storing information. RNA is sort of communicating this message to protein. What I am going to tell you today is a story where this RNA also has now the property to control the protein. ये कुछ नया ही होने वाला है. Okay? I also talked about regulation. मतलब it's not about whether you have a car or you don't have a car. When do you decide to go to BKC? Not during traffic time, right? Or when do you decide to park it inside versus outside? When it's going to rain, you want a free car wash, right? So these are the fine tuning that you can do with machinery so that you get some better outcomes. So here in the case of the cell, it's not enough to say that I have DNA. Is the DNA accessible for me to read? By me, I mean molecules in the cell. For example, there are situations where the DNA is not going to be accessible, and in which case the rest of the process will not happen. One way of regulation. As you can imagine, every single one of these steps is controllable, is regulatable. And together, all of these have been called the catch-all phrase of gene regulation. Are we on the same page? Any questions? All right. So what is new about the Victor Ambros and Gary Ruvkun Nobel Prize, right? The microRNA prize. I want to read this quickly. Once we thought we understood there was all to know about how gene expression is regulated. A cell can tinker with the expression given of a given protein's messenger RNA by modifying the activity, the abundance, or the type of proteins in the nucleus, or with the stability of the RNA once it's made. Everything that we just talked about here. We can change each of these and regulate proteins. But then came a surprising story about a short RNA in the worm, in C. elegans, called Lin4, which didn't encode a protein, but prevented expression of the protein encoded by another gene through something. Okay, this is the surprise, that there is an RNA that is controlling the protein somehow. Okay, so this discovery was made by two scientists, but not in isolation. There is a lot of people who are working in this field, and some of the places that have been involved have been mentioned here. So this is a personal connection for me. I did my PhD in UMass Worcester, and so I was 2 doors down from Victor's lab where all this exciting stuff was happening. Gary Ruvkun is still at MGH and Harvard, and they both studied at Harvard and MIT. So I've kind of geographically located where all of this is happening. So now we will talk about the discovery, the scientists, what systems they used, what were their observations, what experiments did they do, and why did they immediately not see the impact? Why was the prize a bit delayed? Okay, so the first person is Victor Ambros. It's always important to know a little bit of the background of the person who's actually done the work, right? Because that gives you some context of where they're coming from, what challenges they faced. And sometimes, I don't know, I often like to see the path that they have taken, and it sort of inspires me. So Victor actually, his father immigrated from Poland after World War II, and he grew up on a farm, very unacademic upbringing, but he was an intensely curious person. And this is all taken from his interview and a few other biographical sources. He has 8 siblings. Most importantly, he is a first gen scientist, which means that his parents didn't complete college. And this is crucial because he says in his interviews that he wanted to please his dad because he was getting opportunities that his father did not have. And I feel like a lot of us can identify with that. Interestingly, his first foray into science was astronomy. He apparently built telescopes from using books. I don't know how that's possible. Thankfully, we got him into biology. He took a class at MIT when he was an undergrad, and he really got interested in genetics. After that, he worked as a technician. This would be kind of like an internship in David Baltimore's lab at MIT, and then landed up doing a postdoc with Bob Horowitz at MIT, which is where he's going to meet Gary Ruvkun. So that is critical. Gary has had a much more colorful life, if you want to call it. Okay, he was, as a kid, he was fascinated by space launches. Who isn't, right? This is incredible stuff. I was very amused to see that he is also into ham radio, and apparently this is making a revival now. So these are amateur radios that people communicate with, and that's how he learned about circuits. And I think also that inspired him to study biophysics in UC Berkeley. Now this was early 1970s, and so there was a lot of turmoil from the Vietnam War and so on and so forth. So see, yes. So the question is, what is a ham radio? So it is an amateur radio that you can tune. I don't know anything about it because I haven't actually used one, if you can believe it. Maybe Arnab can answer the question.
Yeah, so basically, I just shout so that your mind picks it up. It's a small radio transmitter and receiver that you can build, and you operate your own radio station, your own personal radio station. Usually, you have a certain license that allows you to do it, and you communicate with other such people around the world. And these networks are extremely useful, especially in times of some calamity happens, power is gone, your central network is gone. These ham radio operators can communicate with each other and pass messages and all. It's been a very, very basically a predecessor of walkie-talkies. It's like a long distance walkie-talkie over thousands of kilometers. Let's get to today.
We can discuss this later. Okay. So I like that he said that he marched in protest of the war, but he also went to classes because he was a nerd and he completed his degree. But after this point, I think he was really questioning what he's doing. So he did some off the beat things. He went tree planting in California and Oregon. He traveled south all the way to Bolivia. And there he somehow landed up reading Scientific American, decided that he wants to do science for a living. And because he was bored, he applied for a PhD to Harvard. I mean, that's funny. Anyway, he mastered a bunch of really important techniques that would be useful for the discovery that we are talking about, molecular biology, but we don't need to go into that. Importantly, this is where he met Victor Ambros. So now that we know who has done the work, let's see what they've actually been doing. Okay. The key here is that they were studying all of this, what we're going to talk about, in a roundworm. And the scientific name for that is C. elegans. You don't need to know that. But the key point is that it can be easily grown. It is transparent, almost semi-transparent. You can see inside without doing much. And so, because it's easy to grow, what do? You have a plate on which you have some food, और उसके ऊपर worms डाल दो, like this, and that's it. They survive and they grow. So they are going to now be imaged. And so what you're seeing over here is a particularly unique feature of why people use this roundworm. ऐसे ही नहीं कि earthworm उठा के we can start, right? There's some sense. I already told you this is transparent, so it's making it easy to see. But the key of using a roundworm is that the cell divisions that go from a zygote. Now remember, zygote is when male gamete and female gamete fuse and they form like the first cell. And now that cell is going to undergo lots and lots of divisions to form the adult. Right? We are very clear on this. So an egg fuses with a sperm to get a zygote, and that is going to ultimately lead to a fully functional adult worm. But kya hai? In a normal cell, when you divide, it's not always guaranteed that this side is going to be the anterior and this side will be posterior. But in this worm, it is invariant. मतलब ये बदलता ही नहीं है. It is same. मतलब I can predict. Do you notice that this division is not exactly half? थोड़ा इस side कम हुआ है ना? This is a smaller, this is a bigger cell. This is an asymmetric division, which is how it can maintain a certain polarity. And the beauty is that I know I can map what this cell is going to ultimately become. Matlab, I can say that this cell is going to give rise to all my head cells, for example, in the future. How? Because I can track uska progeny yahan hai, dekho, iska progeny yahan hai. So then these two will become 4, so on and so forth. So these are the stages. You can almost see a worm in an egg about to hatch. Is this clear? This is super important because if you didn't have the predictability of being able to tell कौन सा cell क्या बनने वाला है, so then you don't know which cell type is missing. Remember earlier we talked about cell types, liver and skin. This is not the way this happens in humans, by the way. It's not so fixed. So this is a cool feature of this particular worm. Now I want to talk about the experiment that they kind of did. And for that, I need to talk about something specific called genetic screens. Basically, what we are trying to do is we are trying to understand the function of an organism, the function of something here, by breaking stuff in it systematically. Okay, so I want you to do an exercise with me. Let's say I want to apply the same approach and understand how a car works. Now, all of you know how a car works, largely speaking, right? So I will break one part at a time and I want to see कि car turn होता है या नहीं. Okay? So what if, this is an easy one. In a car, if I remove the steering wheel, will the car turn? This is an easy one. No. Barabar. Without the steering wheel, car should not turn. Right? So this is kind of like what you would be doing if you made a deletion mutant. You have removed something from your genome and now you're checking, okay, मैंने steering wheel निकाल दिया, अभी क्या होता है? Does it turn or not? What if I do something slightly more drastic? If I remove the petrol tank, will the car turn? Some of you are not very sure. Car will not turn, right? Is it because the turning mechanism is defective? No. It's because something even worse is problematic, right? Petrol ही नहीं है. How will the car go anywhere? This is an example of deleting an essential gene where you can't even study the actual process that you're trying to study, right? So we don't want to do something as drastic as remove the petrol tank. If you want to study how a car turns, that is my stated goal. Now this is a tricky one. What if I fix the steering wheel? I don't allow you to turn, but steering wheel hai. And now I ask you to drive the car. Will it turn? No, steering wheel is there, but it's not turning. It's jammed. It will not turn. This is different from this. Remember, structurally it looks like अरे steering wheel है, turn तो करना चाहिए, लेकिन नहीं हो रहा है. Why? Because there is probably some finer problem. These two are very different types of situations. Okay. And finally, this is the most interesting one. मैंने एक आगे का टायर हटा दिया. I know, very horrible situation. And now I drive the car. Now, I'm not an expert, but I believe even if you don't try to turn, it's going to turn because there is no tire on one side. बराबर? ये तो कुछ नया ही हो रहा है. I'm not even turning the steering wheel and the car is turning. Right? This is a new function, a gain of function. जब भी नहीं करना चाहिए तभी भी turn कर रहा है. Right? So, don't worry if you didn't get into the details of the mutants and all. My main point here of explaining is that if I wanted to break one thing at a time and check if the car turns, one function I'm checking, is it guaranteed that the same wrong thing is going to give me the same result? I've shown you 4 different ways in which turning is somehow not happening. But they are very different problems, right? You agree? So in the same way, let's go back to what these guys have done, right? They were looking at the worm to find mutants that cannot lay eggs. They're like, ये क्या बोल रही है? ये क्या हो रहा है? Right? So I want to show you a video, very short clip of how a normal worm, the one that we were just looking at, how it lays eggs. So you can see each of these circles are eggs and one of them is going to pop out. Yeah, that's it, right? Nothing very dramatic, but it's a beautiful thing because there is a vulva, there is an opening through which the egg can come out, and this is the normal process. Now, they use this study they use this phenomenon to find out mutants that cannot lay eggs, just like our car wala analogy, cannot turn. But now you can start to appreciate that there could be many things wrong because of which it's not able to lay eggs. Not just कि भाई ये बंद हुआ. That could be one reason, right? What are the reasons? There could be the one I just described, developmental. मतलब यहाँ पे opening ही नहीं है, no vulva, जिसके वजह से egg बनेगा भी तो बाहर नहीं आएगा. This would be a egg laying defect. The second one is that उसको समझ में नहीं आ रहा है कि अभी egg lay करना है. Signaling problem है. Okay, that means everything structurally is fine, just like how the steering wheel was there, but it was not, it was jammed, it was stuck. So structurally, if it looks perfectly fine, but it's still not able to lay the eggs, that might suggest a different kind of issue, a signaling problem. And the reason that Bob Horowitz decided to do this screen is because this is a very easy to screen phenotype. This is an effect that you can see easily under the microscope and check ये अंडा अंदर है या नहीं. And more importantly, the C. elegans, the roundworm, is not that far away from humans. So studies that we do in this worm, in this simple worm, can actually translate quite well to human beings. So whatever you find there is likely to be very important and conserved. That's the idea. When anybody tells you they're doing research on model organisms. Any questions so far? All right. Oops. Okay. So which mutants did they find? We are only going to talk about these two actually. So this is how a normal worm looks. You can see that it's kind of dark inside. Egg laying defect. मतलब क्या? अंडा अंदर ही है. There is no opening. This was one mutant that they found. One defect. And another one, which we will not go into very much at this point, is some slightly small looking worm, right? So compared to a normal adult, you can agree that this is chota, poorly formed adult. And they actually, the discoveries that they made were trying to figure out how these particular genes worked, because actually it turns out that these phenotypes are quite related. These effects are very related. Okay. Another very curious thing is, for the hardcore biologists in the background, they only found one allele in their genetic screen of the LIN4. Okay, only one defective thing, suggesting that this is probably very small. Whereas they found many alleles of this LIN14. We'll come back to this later maybe. Okay, so you've all understood, hopefully, that there is a mutant, a defective car, right, where eggs are accumulating in the body. And now we are trying to understand कि भाई ये क्यों हो रहा है? What is the identity? What have I actually mutated in this particular organism to get this particular effect? मैंने आपको गाड़ी के केस में क्या बोला था? I am telling you steering wheel के साथ कुछ करना है, because I know the steering wheel is responsible for turning. But if I don't know anything about a car and I'm randomly making some mistakes here and there, then I will not know what exactly I have hit to make the car not work, right? This is what that is. I have an interesting looking worm, but I don't know what has actually caused this particular defect. And that's what this story mainly focuses on. So what is, at the molecular level, what is LIN4? This is the gene that Victor Ambros worked on largely. So let's talk about that. You probably already know this, but this is an RNA, as you can guess, because remember the RNA, small RNA that could, it's in the title. So this is some image, we don't need to get into it. What I want you to focus on is that there is a specific band at a 22 nucleotide length, at a specific length. Okay, there is nothing, no other bands here, but anyway, let's not go there. These are the two people who have actually done the hard work of characterizing what Lin4 actually is molecularly. So Rosalind Candy Lee is Victor Ambros' wife, and she has been a long-term collaborator and technician in his lab. And Rhonda Feinbaum was a postdoc at the time. And so this was a project that both of them really did a lot of the hard work. And what was that hard work? Basically, because they're trying to identify kahan pe galti hua hai in the genome, because of which we are getting this egg-laying phenotype, they made a lot of different mutants to try to understand where this exact mistake is. And because it's such a small answer, right? It's a very short piece of RNA, 22 nucleotides. They had to do many, many more experiments that you normally would. So to give you a sense of this is from their own a paper that they've written about their experience. This was pre-human genome, like a lot of genome sequencing and all. This is pre-classical cloning. And so they have actually sequenced their mutant worms by hand using 18-inch gels. This is a tremendous amount of work. If you don't know anything about this, take my word for it. Okay. And for example, if I wanted to ask, given a certain sequence, where else, what other organisms does this sequence have? In today's day and age, I can put it in some algorithm and get the answer in 30 seconds. But for them, it took almost 3 to 4 months. So it was a very intense process, but they have still managed to get the answer. How are we doing on time? Okay, not bad. Okay, let me cut to the chase because I don't know how much of the technical stuff you might be following. But the short answer is Lin4, the part that Victor Ambros was working on, was actually encoding a very small RNA, and he called it microRNA. Whereas Gary Ruvkun was working on its related gene called Lin14. Now that was the chotu worm that we saw. That was doing this standard thing that we are used to. It makes RNA, it makes protein, right? But the amazing thing that they noticed was that the sequence of this microRNA was complementary to a sequence within this mRNA. Matlab kya? In the Lin14 mRNA, in this part, not the part that makes the protein. This is the part that makes the protein, the dark blue. Okay. This part makes the protein. The part that does not make the protein but is still part of the RNA, the mRNA. Ye laal wala jo hai, the Lin4, will go and bind to it. Okay. And in this particular way, it's able to control whether this protein is made or not by binding to the RNA. And this was a very, very new method. Yes, sir. It is, yes, yes. Okay, so the thing is that they were in close contact with each other throughout because they have been working on this from Bob Horowitz's lab. And so when they got the sequence after doing all this hard work, they actually called each other to confirm. They were reading out the sequence to see whether it is matching or not. And it turns out that it was. Okay. I don't think we should go into this. But the bottom line is that here a small RNA is regulating the levels of protein by binding to the mRNA. Yes? All right, we'll get some questions going later. But the point is, even at this point, this was considered even by Victor Ambros to be something that is likely specific to the worm, कि ये कहीं और नहीं मिला है homology search करने के बावजूद. So maybe this is just a peculiarity of this roundworm. It's not really significant, but he could not have been more wrong actually, because eventually studies found out that there was tremendous amount of क्या हो रहा है? हाँ. So in biology, if a gene has an important function, it's often seen that it's conserved in evolution, meaning the same thing will appear in small organisms like yeast all the way to humans. And that is exactly what a little bit more searching told them. So the original gene that we are talking about is LIN4, and the corresponding genes were found in fly, mouse, humans, so on and so forth. There was yet another gene that behaved in the same way. This bind karta hai to the complementary sequence. So now people started to get a sense ki bhai, this is not just a fluke. This is happening again and again. So maybe there is some importance. So Gary actually is credited with finding this conserved molecule, LET7, forget the name. But the point is, look at the range of organisms that it is present in, suggesting that it is an extremely important and way of controlling genes. A short, you might be wondering कि ये छोटा सा RNA कर क्या रहा है? How is it actually making it so that the protein is not expressed? So the thought is that this particular RNA is binding to a region and preventing certain other factors from binding, and that actually will destabilize the mRNA. So the mRNA levels indirectly will go down. Okay. So the other cool thing that is key about microRNAs is that now we have a lot of evidence that some of these are being secreted out by the cells, extracellular. मतलब वो अपने blood में घूम रहा है. Not just one, by the way. I should emphasize here that there are thousands of microRNAs now that people have searched, and a lot of work has been done. And this is such a conserved mechanism that we can, for example, because these are circulating in the blood, they have been proposed to be used as biomarkers for disease. Because the thought is, in cancer, there are a specific set of microRNAs that are present or absent, and that can be detected in a slightly more non-invasive way compared to what was previously done before. The other complication or complexity, depending on how you want to call it, is that अभी हमने क्या देखा था? One microRNA is binding to one mRNA and controlling उसका level. Turns out that this particular one microRNA has the potential to regulate lots of different mRNAs, क्योंकि as long as that binding sequence is there, it can, in one shot, as a master control, inhibit a whole bunch of other. So if I want to quickly shut down an entire program, that's one way to do it. The converse is also true. One single protein making molecule can also have multiple microRNA sites. So there is a lot of modularity and a lot of interconnectedness in both of these parts. Oops. Maybe this is the slide I'll stop on. So you may have heard of some of these RNAs before, or maybe not, but I just want to clarify this near the end of the talk. What we normally hear of are the RNA that make proteins, right? These are called messenger RNAs or mRNAs. Don't get scared by all these names. The point is that there are so many different flavors. This one is what we are familiar with to some degree. However, mRNA and protein coding genes are only about 3% of the human genome. majority are actually non-coding. मतलब protein नहीं बनता उसमें से. All of these are in the non-coding category. So the one we have talked about today is microRNA, which regulates gene expression. And if you notice, that this binding has some missing part. ये बीच में bubble आ गया है. This is actually a hallmark of the way this binds. It's not entirely binding complementary. Beach mein there is a gap, like in the middle. This is in contrast with the second type of small RNA called siRNA, which will bind complementary. And the reason I bring this up is because these two are very similar, yet they both got two separate Nobel Prizes. So the Nobel Prize for small siRNAs, which are often RNAs that are injected foreign from outside. And the way they were discovered is that the cell perceives this as a foreign DNA and it needs to chop it up because it's a defense mechanism. As opposed to the microRNAs that we were just talking about, which the cell only is making. वो foreign नहीं है. It is using those small RNAs to control the protein. Very different outcomes, but very similar in many other ways actually. And so this Nobel Prize was given to Andrew Fire and Craig Mello in 2006, and it has been used to do many different types of therapeutic applications and all are there. We probably don't have time to get into it, but this is a key difference that I would like you to remember between microRNAs that normally endogenously regulate gene expression in the cells versus siRNAs that are injected. Usually they are foreign DNA, and what they do is this cell is typically going to chop up and destroy things that it binds to because it recognizes it as a foreign entity. I want to just skip to the last fun part, right? So Craig Mello was also at UMass when he got the Nobel Prize. And here he is introducing Gary Ruvkun and Victor Ambros at UMass when they got the prize. Fantastic selfie. But I was very amused to see this, that both of them were actually on his thesis committee. And they were saying that we are still waiting on the revisions for that PhD thesis. So anyway, also Gary is a big ice cream aficionado. He serves ice cream on and off from an ice cream truck that is close to his workplace. And here he is seen serving ice cream to his PhD and postdocs on the day that he got the prize. And I think there are several labs in India that are doing microRNA research for those of you who are interested in learning more. I will close by summarizing what we have talked about today. If there's one thing you take away from today's talk is that there are small microRNAs that are made by the body, endogenous, made by the body. Their length is very specific, 22 to 25 nucleotides long, and their job, their role, is to control the expression of protein coding genes. This is a new way of controlling genes, which is why this was recognized with a Nobel Prize. It's important to remember that the investigations that led to this prize were done in a simple roundworm. Often something we take for granted. We often think, why are people doing research in these model organisms? This is why, because there is so many more powerful things you can do there. The other thing is, this is all curiosity-based research. They were not trying to solve cancer. They were not trying to develop a drug. They were just curious, why is this worm not laying eggs? It might seem a bit silly, but the lesson here is that you never know where basic research will take you. and it can take you to really fundamental, amazing biology, which can in some ways also may be therapeutic. One of the key reasons both Gary and Victor attribute to making this discovery is because they worked together, because it was so key. You can imagine if one is binding the other, you need to know how the story is developing from the beginning. And finally, this is unlike what Victor thought in the beginning. This is not something that is unique to worms. It is seen from yeast to mammals. The little RNA that could, has had a huge impact on the field. There's been almost 25,000 papers published on microRNAs after its discovery, and it's just growing. So with that, I will stop and take any questions.
So first, let's just ask the online audience again.
Should I stop share? Okay.
I mean, if anybody has a question, you can start.
I think there was a, yeah.
Yeah, so please.
Do you want to moderate or?
Go ahead, please ask.
So, I had seen on a slide where there was all types of RNAs. I had seen another kind of RNA which controls and which regulates gene expression, that LNCR. What was that?
This one?
Yes.
So this is long non-coding RNA. We can talk about it a bit later because I would have to go into some more detailed explanation. Yeah,
okay,
there is a question.
I'm going to try and read it out. Can we consider a possibility of spliced out introns being involved in a similar RNA/DNA binding mechanism of gene regulation, or are they too short-lived to have any potential activity?
Okay, let me rephrase the question a little bit. You might be asking if introns do something similar to what I just described, and what I didn't explain today is actually microRNAs largely come from introns only. So the majority of them actually live within genes. So I think that should answer your question.
What are introns?
Good question. I have a slide for that, but maybe.
That's another session.
No. I have but maybe we can take it after the first round of. Okay, I think a lot of people have this question, so let's. No, I'm prepared for this because this is an important concept. 1 second. Don't be scared by the slide I show you, okay? Okay. Oh no. Share. Okay. So normally this is the DNA. Okay. This is the DNA. This is a long thread, almost a long string. I switched. So it's a long string, and I don't know where to start reading for a given gene or where to stop, right? That's a different problem. But suppose I know, okay, a gene begins at this point and ends at this point. So what do? That's the start and that's the end. But you can see that there are these 2-3 colors that are, what am I trying to show you? So in a eukaryotic cell, any one of us, right? The DNA is interspersed, matlab alternating with exon and intron. Okay. What is the significance of this? If you look from the point of view of what makes the protein is made by only the stuff that is blue. The colors are all messed up, sorry. So imagine that these are blue boxes. I apologize. But only the blue boxes will make it into the final mRNA. The other stuff is actually removed. It is cut out and joined. Okay, so what's going to happen is this part will be removed. यहाँ पे एक cut होगा, यहाँ पे एक cut होगा, ये यहाँ पे चिपकाया जाएगा. This same way. So every single blue will get joined to the other blues, simplistically speaking. Now, why is the cell going through all this trouble? इतना क्यों करना है? This process is because I can choose, for example, in a specific cell type, to not include one of the exons. Because now that is going to be a cell type specific protein. जहाँ पे मैंने एक और तीन ही include किया है, दो को include नहीं किया है. So that now gives me some additional modularity on how to make newer and newer proteins, because now you imagine how many combinations can I get. I can either have this one alone, just the second one alone, just the third one alone, or 1 and 3, 2 and 3, right? Or all three together, just with the same piece of DNA. This is very economical from an energetics point of view, right? So if I have control over which pieces of DNA I keep and which ones I throw, then I can make a whole wide variety of proteins. And that process of removing certain regions from an mRNA that has been made from a DNA is called splicing. Because the thing that you want to make into protein should not contain any introns. Okay? Does that? All right.
Okay, I think we can enjoy our chai without knowing that there were introns and all that. Let us do one thing. Let us thank Mayuri.
Thank you.
For taking us through this. Remember that all of you here can come back and ask for. Can you just stop sharing?
I stop.
So all of you here can come back and ask more questions. Online audience, just to let you know, our next sessions, 15 days, 14 days from now is going to be on the Chemistry Nobel Prize at Ruvkun College. Following that, a month from now here is going to be the Physics Nobel Prize. So the next two sessions are on those. You'll find out on our webpage and Instagram, Twitter, all that. Okay, so on that note, thanks Mayuri. Online audience, if you have questions, remember you can always send us an email. We will try and route it to the speaker and they can get back to you. Otherwise, we will stop the session, online session right now. Audience here, let's go down, have a chai and come back and we can ask more questions about everything.
Yeah.
Okay, thank you.